Hello guys, can you hear me? Hello, hello, hi, hi guys, hi guys, hello. I guess I'm live. So today we are going to talk about the Neisseria meningitidis. Okay. So yesterday we talked about the Neisseria gonorrhea. So today we are going to talk about the Neisseria meningitidis. Okay. So let's begin. So this was the yesterday's topic, Neisseria meningitidis. So yeah, we have covered this. Okay. So today we are going to talk about the meningitis. So antigen structure, at least 13 serogroup of meningococci have been identified. Okay. 13 serogroups or 30 serogroups. Meningococci have been identified by the immunologic specificity of capsular polysaccharides. The most common, most important serogroup associated with the disease in humans are A, B, C, X, Y. It's important. A, B, C, X, Y and W135. Okay. So the group A polysaccharide is a polymer of an acyl manosamine phosphate, and that of group C is a polymer of an acetyl O acetyl neuramic acid. Okay. Meningococcal antigens are found in blood and cerebrospinal fluid of patients with active disease. Okay. Outbreaks and sporadic cases in the Western Hemisphere in the past decade have been caused mainly by group B, CW135 and Y. Okay. Uh, Outbreaks in southern Finland and Sao Paulo, Brazil were caused by group A and C. Outbreaks in New Zealand have been caused by a particular B strain, and those in Africa were mainly caused by group A. Okay. Group C and especially Group A are associated with the epidemic disease. Okay. The outer membrane proteins of meningococci have been divided into two classes on the basis of molecular weight. All strains have class 1, class 2, and class 3 proteins. Okay. This is important. So they are divided into classes on the basis of molecular weight. Okay. All strains have class 1, class 2, class 1, class 2, and class 3 proteins. They are analogous to core proteins of gonorrhea. Okay. So do you guys remember what is the core proteins? Core proteins. Do you remember it, guys? Do you remember the core proteins present in the gonorrhea? That is the Neisseria gonorrhea. Yeah. So yeah. So core protein actually prevent the phagolysosome interaction. Okay. Phagolysosome interaction, they prevent better from the interaction of the phagolysosome. Okay. So they are responsible for the serotype specificity of meningococci. These proteins play a role in attachment. Okay. Mhm. As many as 20 serotypes have been defined. Serotypes 2 and 15 have been associated with epidemic disease. The OPA class 5 protein is comparable to OPA of gonorrhea. Okay. Meningococci are pileated, but unlike gonorrhea, they do not form distinct colonies like types indicating pileated bacteria. Okay. So what we can infer from this information is that the we can the gonorrhea and meningococci, they both have pilia. Okay, they both have pilia, but the thing in the meningococci is that they lack di- distinct colony types. Okay, see here. So meningococcal LPS is responsible for many of the toxic effects found in meningococcal disease. Okay, the highest level of endotoxin measured in sepsis have been found in patients with meningococcemia 50 to 100 fold greater than with other granular. Okay, highest level of this is important. So highest level of endotoxin measured in sepsis have been found in patients with meningococcemia 50 to 100 time folds greater than with other granular infections. Okay. So pathogen and clinical findings. Okay. So humans are the only natural source host for whom meningococci are pathogenic. Humans are um, the main animal. Okay, so the nasopharynx, the nasopharynx is the portal of entry. Okay, ठीक है. There the organism attached to the epithelial cells with the aid of pili. They may form part of transient flora without producing symptoms. Okay, or symptomatic. Okay, from the nasopharynx, the organism may reach the bloodstream producing bacteremia. Okay, the symptoms may be similar to those of a upper respiratory tract infection. Okay, fulminant meningococcemia is more severe, with a high fever and hemorrhagic rash. The patient may have disseminated intravascular coagulation and circulatory collapse. Okay, symbol. Mhm. Actually, this whole paragraph is important. Let's highlight it with the yellow color. So, meningitis is the most common complication of meningococcemia. It usually begins suddenly with an intense headache, vomiting, and stiff neck, and progresses to coma within a few hours. Okay, so this is a very dangerous disease, meningitis, which means inflammation of a meninges, the covering of brain. Dura matter, pyometra, and uh, arachnoid matter. Okay, these are three, coverings of the brain. So, so So these three might get affected by the in the meningitis. Meningitis means inflammation of meninges. Okay. So uh, okay, meningitis is most common. Sorry, only. Okay. So during meningococcemia, there is thrombosis of many small blood vessels in many organs with the perivascular infiltration and petechial hemorrhages. There may be interstitial myocarditis, arthritis, and scoliosis. Okay. In meningitis, the meninges are acutely inflamed with the thrombosis of blood vessels and exudation of polymorphonuclear leukocytes, so that the surface of brain is covered with a thick purulent exudate. Okay. So, okay. So it is not known that what transforms an asymptomatic infection of the nasopharynx into meningococcemia and meningitis, but this can be prevented by uh, specific bactericidal serum antibodies against the infecting serotype. Okay. Neisseria bacteremia is favored by the absence of bactericidal antibodies IgM and IgG. Okay. Inhibition of serum bactericidal action by blocking Ig antibody or complement component deficiency C5, C6, C7 or C. Meningococci are readily phagocytous in the presence of a specific opsonin. Okay, okay, this is important. So, Neisseria. Diagnostic laboratory test. So, specimen of blood are taken from culture, and specimens of spinal fluids are taken for smear culture and chemical determination. Nasopharyngeal swab cultures are suitable for carrier surveys. Puncture material from petechia may be taken for smear and culture smears. Gram-stained smears of the sediment of centrifugal spinal fluid or of petechial aspirate often show typical Neisseria within polymorphonuclear leukocytes or extracellularly. So, nasopharyngeal swab culture EM. Okay. Hmm. So, culture media without sodium polyanethyl sulfate are helpful in culturing blood specimens. Cerebrospinal fluid specimens are plated on chocolate agar and incubated at 37 degrees Celsius in an atmosphere of 5% carbon dioxide. Okay, 3 freshly drawn spinal fluid can be directly incubated at 37 degrees Celsius if agriculture media are not immediately available. Okay. Modified MTM with antibiotics, vancomycin, cholesterol, and amphotericin favors the growth of a Neisseria, inhibits many other bacteria, and is used for nasopharyngeal cultures. Presumptive colonies of Neisseria on soil media. Okay. Soil media will time. Okay. Particularly in mixed cultures can be identified by Gram-chain and oxidase stage. Okay. Spinal fluid and blood generally yield pure cultures that can be further identified by carbohydrate oxidative reactions and agglutination with the type specific or polyvalent serum. Okay. Serum. So antibodies to meningococcal polysaccharide can be measured by latex agglutination or heme agglutination test or by their bactericidal activity. These tests are done only in reference laboratories. Immunity. Immunity to meningococcal infection is associated with the presence of a specific complement-dependent bactericidal antibodies in the serum. These antibodies develop after subclinical infection with different types of strains or injection of antigens and are group specific type specific or both. Okay. So the immunizing antigens for groups A, C, Y, and W135 are capsular polysaccharides. For group B, a specific antigen suitable for use as vaccine has not been defined. However, group B vaccines culture from vaccines with mixtures of antigen have been used in many parts of the world. Okay. Currently, there are three vaccines against serogroups A, C, Y, and W135 available in US. Okay. So, a polysaccharide treatment vaccine, Menomun Sanofi Aspir, in which each dose consists of four purified bactericidal capsular polysaccharides, poorly immunogenic in children younger than age 18 months, does not confer long-lasting immunity and does not cause a sustainable reduction in nasopharyngeal carrier. Okay. A, a, a tetravalent conjugate vaccine approved in 2005, Menetreka TM Sanofi Pasteur, is licensed for use in 9 persons in person. Okay, for use in persons 9 months to 55 years of age. Okay. It contains Klebsiella polysaccharide conjugated to diphtheria toxide. Menveo is another tetravalent conjugate vaccine in which ACYW135 oligosaccharide is conjugated to diphtheria. CRM197 Novartis inclusive. This vaccine is approved for use in in in, in individuals of 2 to 55 years of age. Okay. So the advantage of this vaccine is that T cell is that a T cell dependent responses to vaccine is induced. Okay. So this enhances primary response among infants and substantially reduces asymptomatic carriage. Routine vaccination of young adolescent ages 11 to 12 years become before high school using the conjugated vaccine is now recommended. Okay, so vaccination is important in this Neisseria, in this Neisseria meningitidis. Okay. Vaccination is also recommended for persons 11 to 55 years of age who are among the following at risk: person with a functional or surgical asplenia, person with complement deficiency, travelers to highly endemic areas, example sub-Saharan Africa, close populations such as college freshmen living in dorms and military population experiencing a community outbreak and clinical laboratory workers and microbiologists. Okay. So, treatment. Penicillin G is a drug of choice for treating patients with meningococcal disease, either chloramphenicol or third generation cephalosporin, such as cefotaxime or ceftriaxone, is used in persons who are allergic to penicillin. Penicillin. Okay. Penicillin is important. Epidemiology, prevention, and control. Meningococcal meningitis occurs in epidemic phase, example in military encampments, in a religious pilgrims, and in the sub-Saharan Africa, and a smaller number of sporadic inter inter-epidemic cases. About 5 to 30% of a normal population may harbor meningococci, often non-typable isolates in the nasopharynx during inter-epidemic periods. During epidemics, the carrier rate goes up to 70 to 80%. A rising number of cases is preceded by an increased number of respiratory carriers. Treatment with oral penicillin does not eradicate the um, carrier rate. Rifampin 600 milligrams orally twice daily for 2 days, or ciprofloxacin in adults 500 milligrams as a single dose can often eradicate the carrier state and serve as a chemo. Okay, this is important. Rifampin and ciprofloxacin. Okay. Since the appearance of many sulfonamide resistant meningococcal chemoprophylaxis with sulfonamide is no longer reliable. Okay. Clinical cases of meningitis present only a negligible so, present only a negligible source of infection, and isolation therefore has only limited usefulness. More important is the reduction of personal contacts in a population with a high rate cases. This is accompanied by accomplished by avoidance of crowding or administration of vaccines are as discussed. As mentioned, such vaccines are currently used in selected populations, example the military and the civilian epidemic. Okay, guys. So basically, we have finished the mm, live stream and we have uh, done all the works. So yeah, bye guys. Bye bye. Thank you for watching this stream. And tomorrow we will do the all the tomorrow is a special day. Okay, tomorrow we will be doing the gram positive all the bacteria revision. Tomorrow's video will be of like a revision time. Okay, guys. So yeah. So thanks. If you like, then subscribe. Bye. Thank you.